Good morning, everyone. Today I'll be presenting the topic impact of S gene and pre-core mutations on outcome of HBV infection. Myself, Shreyasi Athalye. I am from India. I work at the ICMR-National Institute of Immunohematology. Before moving forward, we have no conflict of interest to declare. This World Hepatitis Day theme was One Life, One Liver, which focuses on the impact that hepatitis B and C silently has on the liver and the effect it has on vital functions of the liver. Now, there are a lot of at-risk populations who are at risk of contracting this infection, which includes healthcare workers, injection drug users, commercial sex workers, tribals, etc. But the household contacts of chronic hepatitis B are one of the most important at-risk group because they are in close contact with infected person. The HBsAg prevalence in India is around 1.4 to 2.7, which is in the intermediate range. However, the low awareness of this infection, poor hygiene, and dense population could put the individuals who are in close contact of hepatitis B at risk of being infected, especially occult infections. Studying mutations in the HBV genome could be one of the crucial ways in understanding the OBI outcomes, and it has been observed in previous literature as well as in our previous studies that all the individuals who are in contact with the hepatitis B case may not present with an infection. Some of them show HBsAg negative, anti-HBC positive, that is a suspected occult infection, and some of them do not show any markers of infection. So these varied outcomes were a question for us. So in the pursuit of that questions, we try to analyze the two important viral genes which were seen in the HBsAg positive individuals as well as the infected close contacts. The aim was to examine HBV in household contacts, aiming to uncover phylogenetic relationships and surface or core gene mutations which could be linked to OBI. Briefly, obtained sera from 22 HBsAg positive family contacts and 90 HBcAb, that is anti-HBC positive family contacts of 62 index cases. We performed HBV DNA detection using nested PCR for S gene as well as the pre-core gene and the real-time PCR using a commercially available kit. The sequencing of the S and pre-core genes was done by the big dye terminator sequencing, and after post-sequencing editing, the analysis was performed to identify the genotypes, the mutations using the Geno2Pheno online tool. The serotyping was done using the S gene sequence using the Purdy et al. Method, and the last one was the PESPA signature, which is available with the LANL database. Further, we performed in silico analysis to identify the physicochemical properties, B cell epitopes, and secondary structure prediction for the HBsAg protein. A brief overview of the results that we obtained. We found that the OBI cases and the index cases had about the same mean age of 35, around 35. The positive members had a lower mean age of 29. The number of males were majority in both index cases and the positive family members. However, in the suspected OBI members, the females were more than the males. Among the 90 OBI cases, 20 showed positivity for HBV DNA, with 14 cases showing positivity for nested PCR, and 66% of these of all the OBI cases showed protective titers for anti-HBs. Looking at the genotypes of the sequences that we obtained, in the HBsAg positive cases, genotype D was the predominant one, whereas in the OBI cases, it was genotype A. Looking at the serotypes, it was evident that ADW2 is the predominant serotype in suspected OBI members, whereas the AYW3 is the common serotype observed in the index cases and the positive family members. Look at the phylogenetic tree. It is seen that the OBI cases grouped with majority of the genotype A, and a few cases, like 3 cases, grouped with genotype T. Mutations in the MHR and C-terminal region were observed, and it was seen that majority of the mutations in the S gene were observed in the MHR region, and most of them were related to vaccine escape, immune escape, or diagnostic escape. Two important mutations in the C-terminal region were found, V194A and S207N, where the latter is a known immune escape mutation, but the former significance is not reported. Look at the RT domain. We found that majority of the mutations were not reported, were responsible for antiviral resistance, but majority of the mutations that were reported did not show any clinical significance. Lastly, we looked at the mutations in the pre-core region, where a majority of the mutations were such that there was no clinical significance reported. The few mutations that were observed were frequently observed either in clinical inactive carriers, were related to changes in antigenic epitope or in development of cirrhosis and hepatocellular carcinoma. We compared the overt and the OBI cases for the presence of important mutations, and these are some of the mutations which were found to be significantly observed in OBI cases as compared to the overt cases. So these mutations were majorly observed in the S gene and the RT domain. The pre-core mutations did not show any statistical significance. The VESPA signature was looked at in the OBI cases that we observed, and it was seen that 6 cases showed V194A signature, and that signature was common in genotype A, whereas when we considered with the genotype D, T127P was the common signature pattern. However, the number of cases were lower in that case. Furthermore, we went to the in silico analysis for HBsAg, where we looked at the physicochemical properties using the protoparam tool. And we found that a few of these mutations were checked for probable effect on function of HBsAg. And we found none of the variants except the stop codon mutation show any significant change in any physicochemical properties. Furthermore, we looked at the hydrophilicity, flexibility, accessibility, beta turn, and antigenicity using the IEDB database, and we found that none of them were affected by any of these mutations that we studied. Lastly, we looked at the B cell epitopes using BC Pred, VIP Pred, ABC Pred, and we also looked at whether the mutations are affecting the antigenicity using Fraxigen, and whether they are showing any IgE epitope using the ALG Pred. And we found that only one mutation, B194A, was affecting the B cell epitope in the C-terminal region from 208 to 110. That epitope disappeared due to after the presence of this mutation, suggesting its possible role in alteration of B cell epitopes. Next, we looked at the effect on secondary structure using the Missense 3D and the SOPMA tools, and we found that there was no structural damage, and also the overall composition of the secondary structures did not change, and the majority of the secondary structure remained a random coil in all the cases. Evidence across the literature shows that mutations in the major hydrophilic region have been associated with vaccine escape, immune escape, and diagnostic escape. Although we have observed some key mutations of statistical significance in S gene, both in the MHR as well as C-terminal region, almost all of them did not cause any changes in secondary structure or B cell epitopes. Only one mutation, V194A, was found to be crucial because it was shown to alter the B cell epitope in silico. However, it was not a statistically significant mutation in our study. Only the presence of a statistically significant mutation may not be able to provide enough information about the overt or occult outcomes. Further in vitro studies are warranted to confirm the role of these mutations in the development of OBI. However, the presence of these mutations indicates the need for a robust screening strategy, especially for high-risk individuals. One of the limitations of our study is that we could not study pre-S1, S2 regions, which are rich in B and T cell epitopes, and which could provide further clues to the OBI outcomes in these cases. To conclude, key mutations related to immune diagnostic escape were observed in the S gene. Antiviral resistance related mutations as well as mutations with no known clinical implications were observed in the RT domain. V194A, although not statistically significant, was one of the important mutations we found that could alter the C-terminal epitopes. And in silico analysis did not show any significant change in secondary structures due to the mutations. With that, I would like to conclude my presentation. I would like to take this opportunity to acknowledge the organizing committee for giving me this opportunity. I thank my department, our director of the institute, Dr. Markaikar, my PhD guide, Dr. Aruna, and a big thanks to my mentors, current mentors, Dr. Anindita and Dr. Naveen, for their confidence in my capabilities. Thank you.